Hi, and welcome to Helomics Corporation. Our state-of-the-art Biosafety Level 2 laboratory is located in the heart of Pittsburgh, Pennsylvania. We occupy over 17,000 square feet and have 4 specialty laboratory areas, including biomarker testing, microarray profiling, live cell assays, as well as a research lab. When specimens arrive at Helomics, they are triaged and appropriately transited to this accessioning space. Specimens are accessioned into our LIS and labeled with barcode stickers displaying our internally assigned identifier. Any paperwork accompanying each sample is also barcoded and electronically indexed. Once logged, samples are transitioned to the appropriate laboratory location depending on the test ordered. For biomarker testing, we start in the grossing hood, where each sample is assessed for sizing, color, and consistency. Cassettes are printed for each sample with the internal identifier and a 2D barcode. The samples are then processed overnight and embedded the following morning to create FFPE blocks. Each sample has a section cut at these microtome stations for H&E assessment to ensure that sufficient tumor is present for testing. When sufficient tumor exists, an order form is provided to the physician, and once returned, additional sections from each sample are prepared for IHC staining and/or mutational analysis.
Each test ordered for a patient requiring IHC staining and assessment is run using one of our 4 Ventana Ultra stainers, or our Benchmark XT stainer. Each drawer on the Ultra system represents a single slide, and all slides and reagents are barcoded for full traceability. All Ventana systems are fully automated from start to finish of staining. During post-staining, slides are washed, cover slipped, and provided to our pathology group for assessment and scoring. We run both lab-developed and FDA-approved protocols and have expertise in method development for novel markers. Our mutational analysis testing is executed in this region of the lab, with DNA extraction from microdissected material occurring in these PCR workstations. DNA extraction utilizes a spin column-based kit. Post-DNA extraction, samples and master mix are plated into a 384-well plate that has been pre-stamped with primers and probes utilizing the Biomarker FXP instrument. The Biomarker instrument includes both a 96-well head as well as a span 8 arm. After plating, the qPCR plate is sealed and moved to the VS7 qPCR instrument for analysis. The VS7 is also outfitted with automation, the Twister Bot, which allows us to load up to 150 qPCR plates queued for analysis. The Biomarker instrument also includes on-deck components to allow for unsealing, sealing, plate reading, and automated thermal cycling. Plates can be transited to these on-deck components by using the servo shuttle. Additionally, on-deck heating, cooling, and shaker stations can be utilized to automate other processes, including our library prep for microarray profiling.
The front end of our microarray profiling assays happen in this section of the lab. RNA is isolated from the samples utilizing a kit-based spin column extraction. Once obtained, the RNA is converted to cDNA, which is subsequently amplified in preparation for addition to an Affymetrix chip. This process occurs in the hoods along this wall. Additionally, the ability to extract nucleic acids from fresh tissue as well as blood is also housed in this region of the lab. The remainder of the microarray profiling assay is completed in this laboratory space. The amplified material is chemically and enzymatically fragmented as well as labeled. The fragmented and labeled resultant material is then hybridized to an Affymetrix array specific to the assay being executed. The hybridized chips are washed and stained using the Affymetrix DX2 fluidics line. Once unbound material is washed away and the stain is amplified on the bound material, the chip is scanned. The scanned information is then funneled into the appropriate algorithm and a patient result generated. The Affymetrix DX2 line, while CE marked and approved for diagnostic use, is flexible enough to run all Affymetrix chip types. This applies to both the clinical and research settings.
Within our live cell section of the lab, we're able to execute the live cell assay on samples that arrive here as fresh tissue from a given patient. Post-receipt and accessioning, samples are processed, minced into small explants, and added to the appropriate number of flasks for growth. Samples are then placed in incubators and assessed on a regular basis for confluency and overall health. Once a sample has reached a sufficient fluency for plating, the cells are harvested and plated into 384 well plates using automation. Cells are then challenged with various chemotherapies. Finally, cells are fixed, stained, and imaged utilizing proprietary scanning systems. These proprietary scanning systems allow us to image each patient case to identify the number of cells in each well after they are fixed and stained with DAPI, a nuclear stain. The resulting counts are assessed to identify which chemotherapeutic agents are more effective for the patient and which are less effective. The live cell capabilities described previously are mirrored within our research laboratory, allowing us to offer that platform to clinical research partners. This suite of equipment provides the capability of performing endpoint functional live cell assays. In addition, a variety of proteomic or molecular testing may take place throughout the course of the sample culture period. Our research laboratory also has a broad range of molecular and proteomic capabilities. Dedicated spaces for RNA and DNA extraction, as well as qPCR analysis, are located here. Any research projects that require microarray profiling utilize the Affymetrix DX2 line highlighted earlier in the tour. This section also houses protein expression analysis modalities, including Western blots and our Luminex instrument. The Luminex FlexMap 3D is a multiplex bead-based protein detection system. This instrument is capable of quantifying dozens of proteins per sample on 96 well plates.
Housed within our laboratory is also a pathology suite. With our board-certified pathology staff, each sample submitted for biomarker testing is assessed and scored for final reporting to the ordering physician. The pathology staff is also available for clinical research projects in a wide range of capacities. This room houses any inventory not yet pulled for use in the laboratory. Overall inventory is tightly controlled by our dedicated materials management group. Our inventory team keeps close tabs on any reagent or instrumentation that arrives at the facility and appropriately triages any of these things that are not validated for use in the lab. These items are quarantined until properly qualified by our lab staff. Additionally, a cold storage room is present on site for bulk storage of cold reagents as well as our internally created FFPE blocks. All cold and warm storage are monitored by Reitze Scientific, the small box on the wall. This monitoring is 24/7, 365, and lab staff are on call for after-hours addressment of any equipment issues. Thanks for joining us on the tour. We hope you enjoyed yourself.
For more information, please visit us at helomics.com.